you're having a great day. Today we're going to tackle chapter 2, which deals with some basic chemistry. I know a lot of people are not the biggest fans of chemistry, but we're not going to in depth, with this information, and we're going to keep it fairly basic, and I'm going to make sure that I'm really kind of focusing on how the chemistry relates to our class, anatomy and physiology, in the human body. So let's go ahead and let's get moving with this information. First off, obviously, whenever we start to talk about chemistry, we begin with atoms. And so let's begin talking about the basics of chemistry, and then let's start to focus on atoms to understand how all of this chemistry comes about. At the simplest level of organization, human body's composed of atoms, and then we put two or more atoms together and we can create molecules. Okay, now these ions that are mentioned, these are something a little bit different. These are positive or negatively charged atoms. So they're a specific type of atom, and we'll talk about those here in just a minute. But again, the main thing that we're going to focus on in this chapter are going to be atoms and molecules. And in the process, we will talk about these ions. In order to truly understand physiology, again, don't forget metabolism is chemistry taking place inside the body. And so to truly understand physiology and metabolism, we have to understand some basics of chemistry. Again, I know most people don't like chemistry, so I'm not going to kind of drown you in information that you don't need. First off, matter. Let's talk about matter. Matter is a good definition of matter. And again, as I go through this information, you can take some notes. It's always a good idea to take notes. You can always pause it, go back a few seconds, and replay it. Hear that again if I'm moving too fast. But matter, let's go ahead and take down a definition of matter. It's anything that has mass and takes up space. And so on our planet, matter are these physical objects, right? These things that we can touch, we can feel. So matter, anything that has mass and takes up space. And so there's three forms of matter: solid, liquid, and gas, that we're going to discuss. Solid, liquid, and gas. Solid is obviously denser molecules together. Liquid is a little bit more spread out, and then gas is even more spread out. So we can think about water as a liquid. If we freeze it, it becomes a little more solid, because those molecules get closer together, and then as we heat it up, it becomes a gas and it evaporates. And so we can see those three forms of matter in water. Again, an atom is the smallest stable unit. Okay, it's the smallest stable unit of matter, really, is what it is. So here it says it's the smallest particle that exhibits the chemical properties of an element. That's a terrible sentence. Let's take down the definition of an atom again. Again, it's the smallest stable unit of matter. If we put an atom on the table, it's not going to degrade automatically into its subcomponents, right, into its subatomic particles. And so it's the smallest stable unit. Now, let's talk about these elements real quick, because this is important. Again, this was in the review. If you didn't watch the review, maybe watching that again, as well as reviewing this this presentation, would be a good thing to get caught up on that chemistry. But an element, all these atoms have different numbers of protons and neutrons and electrons. Now, all the atoms have the same number of protons. We group them together, and they all have similar characteristics. And so we call all of those with the same number of protons an element. So elements are groups of atoms that have the same properties, and their properties are kind of dictated on the number of protons that they have. And we'll talk about that again here in just a minute. Again, I mentioned just a second ago that atoms are composed of smaller particles called subatomic particles, sub below the atom, right? So again, anytime we're in this class, you should be trying to learn how to break words down, so it makes it easier for you to understand words that you might not know that you encounter, in this class. So subatomic, below that atomic level, we've got three of these particles: protons, neutrons, and electrons. Protons have a positive charge, electrons have a negative charge, and neutrons have a neutral charge. That's kind of where that name comes from, right? The neutron, neutral charge. So they have no charge. So again, the number of protons is really what kind of determines the characteristics of that atom. I'm sure that you're a little bit familiar with chemistry, and you've probably heard before that the electrons down here, these guys are orbiting around in a shell, and the protons and the neutrons, these guys are in that atomic nucleus. Now, the electrons we can give up and we can accept. So we can kind of change the number of electrons. So these guys don't really define what the atom is. But what determines the number of electrons is the number of protons. And so the protons, we can't change those. If we change the number of protons, then we get a totally different atom. Okay, if we change the number of electrons, we don't get a different atom necessarily, right? Still the same basic atom, it's just got a different charge to it. And that's really how we create those ions. So protons are truly what give each atom their characteristics, and as a result, they are what kind of create each of our groups of elements. Each atom with the same number of protons equals one element. Now, the protons are always the same number in each atom, so they give a positive charge. So in order to balance that atom out, that atom usually has, naturally, the same number of electrons to balance it out to zero. So the protons actually determines how many electrons is in that atom as well. And as we just mentioned, the electrons are what's responsible for binding and creating our molecules. So let's move forward a little bit. Let's talk about that. Molecules and chemical compounds. First off, whenever we put two or more atoms together, then we tend to refer to them as a molecule. Our textbook likes to also talk about and call them compounds a little bit more, depending on which type of molecule you are. But one thing that's always true is this thing called the octet rule. So the octet rule states that most of these atoms are trying to get 8 electrons in their outer shell in order to not be reactive, in order to be happy, to be just kind of chill, right? So again, these atoms have the same number of protons and electrons. So that is balanced to begin with. That's how the atom comes, right? Protons equals number of electrons. But how many electrons that atom has in its outer shell, if it's got less than 4, it's probably willing to give up those electrons to become more stable, okay, and less reactive. Again, because it's those electrons that determine the reactivity, the ability to bind. So if it's got less than 4, it's more than likely to give those atoms away, those electrons away. And if it's got more than 4, it's more than likely to try to find more electrons to get its 8, right? So you can either give away your extra ones to drop down to the next shell that has 8 of electrons, or you can add to that existing shell, and then you can go up to the next 8. So that's kind of what this octet rule does. This is really what determines how we bind, because we're always trying to get 8 in the outer shell, okay? Now, there's 2 different ways. Let me go back, sorry. There's 2 different ways that we can achieve this, that we can get this 8 in the outer shell. One way, as I just mentioned, is to give electrons away or to accept electrons. So what this does is by removing an electron or adding an electron to an atom, we're changing its charge. And so what we create by either gaining or losing electrons is called an ion. So if we create ions, we create something that now has a plus charge and something that now has a minus charge. And just like magnets, a plus and a minus are going to attract. And so ionic bonds are formed when 2 ions, a plus ion and a minus ion, are just kind of attracted, kind of like magnet poles, right? A positive and a negative pole on a magnet kind of linked together. On the other hand, we have covalent bonds. So co means to share, and valent literally means electrons. And so in a covalent bond, if 2 atoms can hang out close enough together that they share their electrons, they don't have to give them up or accept them, but they can just hang out together and share those electrons, then they're going to be bound by that sharing, and that is called a covalent bond or a covalent molecule. Now, our textbook likes to consider those covalent bonds to create molecules, and so they call usually the ionic bonds more compounds. And so I'm used to calling both of these molecules either ionic molecules or covalent molecules is how I was taught, but this textbook kind of separates it a little differently. Let's talk about ions first. Again, a good definition of an ion is a charged atom. It is an atom with a positive or negative charge. Again, how does it get that positive or negative charge? By either losing or gaining electrons in order to try to hit that 8 in the outer shell. If they've got 1, 2, or 3 in that outer shell, well, if they lose those, then they drop down to their next shell that has 8. Or if they have 5, 6, or 7, if they can just gain some more, then that satisfies them up to the 8 level, right? So ions, we're going to talk about a good bit of ions as we go through this semester and especially next semester. These guys are super important for physiological functions. Things like sodium and potassium are so important for your nervous system, your muscular system to work properly. Calcium as well is super important for your muscles to work properly and your nerves to work properly, as well as clotting. Other ions are listed in the textbook, and we see this table that lists all these ions and gives their basic kind of abbreviation and also kind of tells you some of their importance. Now, this information is not going to be on the test. You don't have to study this and know this, but this is a great reference if you're ever curious. Well, what if I've got too much sodium or not enough sodium? Well, then here we can look and see what it does. What if my magnesium level is low? Why do I feel sluggish? It's because you're not making enough energy. And so we can also look and see there's a few more over here, but these handful of ions, these guys are created by losing or gaining electrons. Okay. So if we lose electrons, for example, here sodium, it's got a total of 11 protons. Okay, so sodium has 11. So it originally has one extra in its outer shell. We didn't talk about this. You don't need to know this for a test, but always the first shell, the innermost shell has 2, and then the rest of them have 8 is the way that we think about it. So we go 2, and now that gives us 8 in the next shell, and so sodium has 3 shells, valence shells, 3 electron orbits, and in the outer one, there's only 1. So if it gives away that 1 electron to another atom, it becomes balanced, it becomes more stable, but now that it's given away a negative, it gave a positive charge. So now sodium giving away the negative makes it positive. We call those cations. I know this looks like cation, like vacation or something, but this term is cation. So the cation is positive. Now, the atom that gained that electron. So here's a great example. Chlorine has 17 protons, and so it's got 7 in its outer shell. It needs 1. So if sodium gives up 1, sodium became positive, it became the cation, but now chlorine is going to gain that electron, and now it gained a negative. So it is more negative charged. A negative charged ion is called an anion. I know it looks like onion with an A, but an ion is a negatively charged ion. Now that we have a positive and a negatively charged ion, they can just attract based on charge. Like, again, the magnet concept. When they start to attract, they create these compounds, and they start to be held together in this lattice work is the way that they like to describe it. Okay. So here again, we've already mentioned this about how many is in the outer shell and what they tend to do, either donating or accepting those. So I'm not going to review that again. But here, something that I do want to mention is that whenever we bind that ion and that anion, many times we do form salts. So most of the time we're going to form some sort of salt. So an ionic compound is usually referred to as a salt, right? And so there's many different salts. It's not just table salt. There's many different types of salt. Okay. Now, here's a good visual. Oh no, this kind of cut some of it off. So here we can see that sodium has one extra, and it donates it over to chloride that has 7 in its outer shell, its outer valence shell. And so now sodium loses its outer, drops down to its next one, which is a happy 8, and chlorine gained 1, and now its outer shell is a happy 8 as well. But now what we've done is sodium has a positive charge because it lost a negative, and chlorine has a minus charge because it gained that negative. And now that we've got these two together, they're going to start to kind of cluster together and form these ionic bonds. Again, it starts to form a crystal. Whenever we talk about salts and ionic bonds, we form a crystalline structure. Covalent bonds are more important as far as physiology many times. And so covalent bonds are what's going to take place whenever we share electrons instead of give them or mix them. And so sharing of electrons, again, the term co means to share and valent refers to electron, forms what they like to refer to as molecules, true molecules, molecular compounds. Okay, so if we talk about making these covalent bonds, we have two different types of true molecules that we can make after that. Okay, now covalent, and we'll talk about that here in just a second. Those are going to be organic and inorganic molecules. But first, let's talk through and a little bit about the basics of covalent bonds. First off, they're created whenever we share electrons, and the 4 most common elements that form these covalent bonds in our body are right here, and these are the most common elements in our body: carbon, hydrogen, and oxygen. Okay, C, H, and O plus nitrogen. Those are the 4 biggies. But CH and O, don't ever forget. These CHO is how I like to remember it. CH and O. These guys are super important to build those organic molecules, and those are the ones that can build life. Here we see a hydrogen atom. It has 1 electron in its outer shell. Well, it can hang out with another hydrogen and satisfy for that inner shell. Remember, that has 2. So H2 is how you might find hydrogen instead of H by itself more often. Your carbon, if we look at this, carbon has 4 electrons in its outer shell. So that means it has 4 spots to bind. So it can bind with 4 hydrogens. And here, if it binds with 4 hydrogens, it forms methane, right? So there is methane. Here we can see oxygen has 6 in its outer shell. So it's got 2 open spots. So it is happy if it just grabs 2 H's. So that's the reason that this is commonly made on our planet, is that oxygen naturally binds with one of the most abundant atoms on our planet, hydrogen, and because it's only got that 1 electron, it's very reactive, and so here it's going to bind, and we get water. Okay, super important. Now, number of bonds on an atom, the number of electrons that it shares determines the type of bond. That's a better way to say it. So you may have heard before about double bonds or triple bonds or quadruple bonds. So it depends on how many electrons it's sharing. For each electron pair that's kind of there, then that equals 1 bond. So again, you could bond more than 1 together. 2 oxygens can bond together, and that's by a double bond. Okay. pH. Let's talk about pH real quick, because this is super important before we get into some of this information about organic molecules, and that's really the most important information for this chapter. pH is a measure of hydrogens, not just hydrogens, but free hydrogens. A hydrogen that is not attached to anything else. It's floating freely in a solution, and so pH is a measure of how many hydrogens are available. Here is the equation for pH. You're not going to have to do any kind of math on the test, but I want you to understand what this equation is doing. First off, right here is our hydrogen. This is the variable. This is what we're measuring, right? So it is a measure of hydrogens. Now, these brackets mean that we're measuring a concentration. And when we look at this concentration is going to be in super duper high numbers. And the reason that we know that is we're using this thing right here, a log. If you've forgotten what a log is, don't worry, I do too. But a log, this is simply a way to take super big numbers and make them smaller, especially so our brain can deal with them easier. So instead of seeing numbers like 10 to the 14th power, 10 with an extra 14s on it, and trying to say that and write that down, we can just say 14 because we use the logarithm. So the logarithm of a big number takes a big number and makes it small, makes it easier to understand. What the log really does is it takes the numbers of hydrogens that are possible in a solution and it breaks it down to a scale of 0 to 14. Instead of 10 to the 0 and 10 to the 14, now we just say 0 to 14. Okay, but here that log is taking that big number and making it small so we can have an easier scale to deal with. Now, what this negative does, this is important, this flips that scale. It makes the inverse of the log for the hydrogen concentration. So the greater the number of hydrogens, the lower the pH. So it flips the scale. Excuse me. The more hydrogens equals a lower pH, and the less hydrogens equals a higher pH. Okay, so what we're really doing here is we're measuring a lot of hydrogens because hydrogens are super reactive. They've only got that one electron, and we're going to measure big amounts. We're going to break it down to a smaller number, but as we have more hydrogens, we actually go lower in the pH. As I mentioned, I'm sure you've heard of the scale 0 to 14 before, and right in the middle is 7, and 7 is neutral. Plain water is at 7. And then as we add hydrogens or take away hydrogens, we go more acidic or more basic. So again, the more hydrogens we have, we're going to start heading down. And you notice we've got a reverse of the exponent here, right? Reverse of that logarithm. And so whenever we look at this, that pH of 1 is super highly acidic. pH of 7, well, that's neutral. Okay, so below 7 is an acid. And again, those are going to have more hydrogens. And above 7 is going to be a base. And again, these guys have less hydrogens compared to the acid side. So here's a little chart just kind of pointing that out. Here's the amounts of hydrogens in the acids versus the bases, right? So you can see more versus less. Here's some of the things that you might be familiar with: tomato juice, grapefruit juice, wine, even beer is down here around this tomato juice range is a little bit higher. It's usually about 5.4 or so. So here we see some of the things. Lemon juice and stomach acid are very similar, right? Hydrochloric acid is your stomach acid. So if it's just pure hydrochloric acid, it's much lower. But look at this. Check it out. That there's other things. The farther we get away from 7, the more damaging it can be no matter what. So things like bleach, household bleach at a pH of 12. It's not an acid, but it's pretty basic. And so bleach can do damage to things. We can see that if you pour bleach on something, it will start to eat a hole straight through it. It will eat a hole through wood if you pour bleach on wood and just leave it there, right? So it's kind of similar to what some of these acids over here can do, all right? Now, buffers. Let's talk about buffers real quick. A buffer is a way to neutralize an acid or a base. A buffer, a good definition, is a substance that resists a change in pH. So a buffer you can add something of a different pH to a buffer and the pH won't change very much at all. And so buffers are very important in order to bind these hydrogens so they don't become reactive. Add that note too. Not only are buffers substances that resist a change in pH, but more importantly, how they do it is they bind hydrogens so they don't affect pH. So we need another chemical that can bind up these extra hydrogens and then be able to almost transport them. That's really what buffers do. In the long run, they're going to bind these hydrogens and they're going to transport these hydrogens to where they can get rid of them. Okay? In the long run, we're going to try to get rid of most of them in the urine. If it's direct hydrogens, we can just add it to the urine and we can urinate it out. As long as there's buffers involved, then it's not going to be very acidic. So the urine, even though it contains a lot of acids, it's buffered and the pH is only about 6, right? So that's a good thing. So again, these buffers, they can help resist that change in pH. And they're also really kind of hydrogen carriers, transporters, so that they don't affect negatively on the body. Now, here's the main thing that we're going to talk about today: organic versus inorganic molecules. Now, the organic molecules are molecules, and they don't really tell you this very well right here, but organic molecules, this is super important. Make sure that you take this note right now. Organic molecules have to have CHO. They have to have carbon, hydrogen, and oxygen, CH&O. If they don't contain all 3 of those, then it is an organic molecule. It cannot produce life. That's what organic molecules do. Organic molecules build our smallest living structure, which are cells. And so if we don't have carbon, hydrogen, oxygen and create these organic molecules, we can't create life. We can't create the cell. We can't create tissues, organs, and eventually that organism. Okay? So these are very important. Now, if you don't contain all 3 of those, if you're missing just 1 or 2, then you are inorganic, right? But inorganics are not useless. Inorganic molecules only contain carbon and oxygen. Well, you made carbon dioxide possibly, right? If you just have hydrogen and water, or excuse me, hydrogen and oxygen, well, you just made water, right? And we breathe in O2, 2 oxygens bound together. That's an inorganic molecule. So inorganics are not wasted molecules. They're not useless. These are still very important molecules to help maintain life, okay? Now, the focus, though, is on creating life. And so we're going to start really focusing from here on out on more of our organic molecules. We do have 4 classes of these organic molecules, and this is kind of out of order of how we usually cover them, but this is how the textbook does it. First, we've got lipids, which are fats and oils. Then we've got carbohydrates, which are more sugars and starches. We have nucleic acids, which we discuss last because these are very specialty molecules. Each one does something very special. Nothing else in the body does it. And then we have proteins, and proteins are the biggies. Proteins create structure, they build structure, and they create function. Okay, so proteins really give us our anatomy, our structure, and our physiology, our function. And so let's finish this discussion by about these for about 15 minutes or so. Lipids. Again, lipids are fats and oils. And let's talk about lipids in general real quick. Lipids are fats and oils. Again, animals tend to make the fats and plants tend to make the oils. If we talk about at room temperature, what is each of them doing? Lipids, the animals, the fat tends to be solid, and the oils tend to be liquid. This is one of the reasons that eating too much fat is bad for your blood vessels. This solid-like substance, it dissolves in your body because everything dissolves when it heats up a little bit, but it still has the ability to form these solid clots in your blood. So again, eating more olive oil than butter is better for you, in the long run. So these lipids, as we talk about them, I want you to know the building blocks of lipids are fatty acids. And so these fatty acids are the real building blocks, but we see right here in our body, this is the basic building block of lipids in our body, triglycerides. So I want you to know 2 building blocks for lipids. This is what we tend to make all the bigger ones out of, are either the main thing are fatty acids, but we can group 3 fatty acids together with another molecule called glycerol, call it a triglyceride, okay? Now, lipids, talk about them as stored nutrients, right? But a lot of us don't unstore those nutrients. We just keep storing more, right? That inner tube around our gut just keeps getting bigger. So, do want to mention some other things. There's some membrane components and also for some hormones, but also want you to know lipids are super important for protection. They're important for padding and cushioning and protection. Adipose tissue, which is full of triglycerides, is really just bubble wrap. It's fat-filled, liquid-filled bubble wrap. That's all that it is. So here we see 4 groups. So let's talk about these real quick. Triglycerides are the first group. Again, this is the long-term energy warriors. If you eat a lot of carbohydrates, you burn energy. You're going to convert that energy to lipids and store that for as long as needed. So storage, we just gain adipose. We don't lose it. And so as a result, we need to store in the form of adipose. That is what our fat is made out of. Fat in our body is just liquid triglycerides inside of these bubbles, adipocytes, right? Now, this is, again, used for cushioning, insulation, protection. We can use it to support structures in the body. But again, it is primarily needed for long-term energy. Now, we've taken ourselves away from this process where we kind of build up our fat reserves before we see lean winners. We see animals doing that, right? So the bears are starting to realize that the fall's coming, and so once it starts to get cooler, they're going to go out. Squirrels, they're going to go out and start storing away in their bodies and elsewhere to make it through the winter. So these guys are really long-term storage, especially for times like that when we're going to hit these lean times during the winter and we need to have this extra energy to burn off slowly. And that's the problem with losing fat, is it takes a long time to burn that fat. But this adipose is there. Once you create it, you can't get rid of it, and it likes to reinflate super quickly. So if it takes a long time to lose that fat, but then it takes a very quick time to reinflate that fat bubble and gain that adipose right back. So that's the reason dieting is so difficult. You really have to make a lifestyle change instead of just getting on some crazy keto diet, some weird diet that really doesn't make sense. Okay. Now, I don't care if you know these terms. We'll talk about those as we get through later in the semester. Okay, this image is kind of messed up, but you can kind of see here is glycerol, and then here is a chain. We can't see the whole chain. Here's a better view up here maybe. There's glycerol, and then there's 3 fatty acids, and that's why we call it a triglyceride. Phospholipids is the next group of lipids we want to hit. These guys are super important for cell membranes, and we're going to talk about that when we get to chapter 4, I think is the chapter for cells, but when we get to the cell chapter, we will talk about that. Again, fat doesn't like water, so phospholipids basically creates a layer that pushes water to both sides of the cell. And so it pushes water towards the inside, and then it pushes water towards the outside of the cell. And this is going to allow the cell to specialize on the inside, undergo different metabolism, and there's what creates life, right? And so these phospholipids, super important to create that phospholipid bilayer, and we will talk about that here in a bit in the next chapter that we discuss. Again, hydrophobic, kind of pushes that water away. Steroids are super important lipids. These are hormones. Okay, so these hormones are very powerful chemical messengers. These guys can roll through our bloodstream, and then they can start to change the actions of other organs and tissues down the line. Now, these steroids are made out of cholesterol. I don't know why they list cholesterol as a steroid. That's kind of stupid. But cholesterol is the main thing that we build steroids out of. And we can also create bile salts from some of these fats as well. But when I say steroids, we're only talking about the hormones, okay? So this is a little weird how they've tried to discuss this. So again, these are very important hormones, very powerful hormones. Usually the sex hormones are the primary steroids that we think about. Icosanoids, these guys are local acting hormones, and so many of these aren't even hormones. Many of these don't use the bloodstream, so they're really more local messengers, but they can act like hormones and they can cause changes in the tissues around them. So anytime we use the word local, that means that it's made and it's used in the same place. It reacts in the same place. So here it's made and used basically in the same tissue. Here's a list of those. I don't really care if you know that for the test. Just want you to know icosanoids, chemical messengers, many times involved with some of the immune system, inflammation, sometimes nervous, but it's just about it. Some of these other lipids, I'm not too concerned about these for the test. Glycolipids on the surface of a membrane help giving it its identity, help it with binding for cellular binding. And then we also have some of these other vitamins that we'll talk about that are types of lipids, but I'm not too concerned with that. Let's now, so here again, here's that triglyceride. Here's glycerol, and here's a long chain. That's a fatty acid right there. Okay, so here you can kind of see the basic formula is cholesterol with these 4 rings, and then we kind of modify it and we can create steroids out of it. All right, not that big a deal. You don't have to worry about that for the test. Let's move on. Let's focus on carbohydrates. Carbohydrates, literally carbon to water. So we just add 1 carbon for every water. 1 carbon, 2 hydrogens, 1 oxygen. So that's kind of always the equation, CH2O. If we've got 6 C's, we got 12 H's, and we got 6 O's. So there is glucose, C6H12O6. Carbohydrates, these guys are super important for energy, right? This is the gasoline that we have to put into our engine in order to create that combustion, which is going to be ATP, which is really kind of moving things in our body, making things work. So these carbohydrates, super important for energy. But again, as I just mentioned with lipids, if you have a bad habit of eating carbs than you burn in a day, well, then you're going to gain fat. Even though you ate sugar, you're going to gain fat. And this is the problem. Carbs are not bad. There's a lot of wackos out there, wacko diet people who say carbs are bad. And that's not a good thing at all. Carbohydrates are important. If you eat a balanced diet, then you should get approximately 65% or so of your calories from carbs. But the point is, if you eat them, you should burn them. And if you do not burn them during the day, then you're going to start to save them and turn them into something that you don't want, which is going to be that adipose. Okay, so limit your carbs and make sure that you are active. That's the key. Most people, they're lazy, so they eat a whole bunch of bad stuff and then they just sit around. They do nothing all day. So you have to, again, change your lifestyle, not go on some wacko diet. All right. The building blocks of carbohydrates are monosaccharides. Mono, again, means one. Saccharide refers to sugar. We also refer to monosaccharides as simple sugars. Now, we can talk about disaccharides and trisaccharides, but then we can just get to saccharides. If we've got more than one monosaccharide together, then we can consider it a polysaccharide. Again, monosaccharides are our simple sugars. This is what our body actually uses as fuel, but we eat polysaccharides many times and have to break them down into monosaccharides. So again, these are our simple sugars, the monos, and our polys are more of our complex carbohydrates, our starches, right? Now, you do need to know, like I said, the building blocks are the monosaccharides, but the most common, and I just mentioned it, monosaccharide that we're going to discuss is glucose. So glucose is it is the main monosaccharide in humans. This is the gasoline that goes in the tank. You don't put anything else in that tank hardly. And so it's the same thing. This is what goes in that creates energy. Even if you're eating a whole bunch of protein, then you're going to have to convert it to glucose in order to get it into mitochondria to really produce that energy. And so glucose is super important. This is the one we're going to talk about, C6H12O6. Go ahead and learn that. We can take long chains of glucose and we can store it in our body. And we store that in this form right here. And you need to know this. This is called glycogen. So glycogen is just long chains of glucose. And we tend to store it in our liver or in our muscles. And that's where we have it ready to be used, especially in the muscles for exercise. Okay. So here's an image kind of showing you one glucose, and then here's trying to show you a chain of glycogen. There are carbohydrates. We're not really going to talk about these too much right now. Again, the disaccharides are just 2 monos together. We'll talk about these more when we get to digestion and metabolism and things like that. Okay. Again, polysaccharides, I want you to focus in animals. As I just mentioned, glycogen is our long stored chains of glucose, but in plants, we tend to see starch. Cellulose as well. That's our fiber. Cellulose is what we refer to as fiber in our diet. But starch, this is potatoes, this is pasta, right? This is bread. So these are those long chains of stored sugars that slowly break down. These are our complex carbohydrates, is what we usually call them. Here's some examples, more examples of monosaccharides. Here we can see galactose. This is your milk sugar, lactose, right? So this is galactose, the milk sugar. There's fructose, more of a plant sugar. Here's some other ones, ribose, deoxyribose there, both of these, DNA, right, deoxyribonucleic acid. Here's some disaccharides, sucrose, lactose. Here's that galactose mixed with a glucose to make lactose sugar. Here's maltose, again, malt sugar, like a malted milk ball or what you make beer out of, right, malt, malted sugars. Next, let's talk about nucleic acids. Nucleic acids, again, these are specialty molecules. That's what you need to know. You can't define them as a group as having a big function. Each one does its own thing. So nucleic acids are specialty molecules. So we need to talk about each one individually. Before we do that, though, I want to flip slides just a little bit. I want you to note the building blocks of nucleic acids are nucleotides. Okay? Some people say nucleotides, nucleotides, whatever, enunciation on the O differently, but it's the same thing. So these are the blocks. I don't really care that it's a sugar, a phosphate, and a nitrogenous base, but that's what it is. These things look like the big letter L, and so this is how we're going to create all of our nucleic acids, is by using these nucleotides as our building blocks. The first example of a nucleic acid, an Na, is DNA, deoxyribonucleic acid. Again, this is the complete blueprint to build any protein in your body. So any protein that you can possibly make, be it structural or functional, is coded in this DNA. And each cell has a full copy of this DNA so that each cell can make whatever it needs to make in the body. Now, this is inside the nucleus. This never leaves the nucleus. There is a different type of it in the mitochondria that we'll talk about whenever we get to cells. We focus on this being in the nucleus only. Again, this is a big code, the big blueprint. We can make a copy of one protein code and take it out of the nucleus and turn it into that protein. And what we're going to use there is ribonucleic acid, RNA. So this is a single strand. It's simply a copy of only one protein code, and it leaves the nucleus and it enters the cytoplasm, and there it can help build it. Now these sound similar, but this is a full blueprint, and this is only a copy of one of parts. And so these are truly different types of molecules. Here is our third, well, here's an image that shows you DNA. But here's a third type of nucleic acid I want you to be familiar with, ATP, adenosine triphosphate. So ATP, or adenosine triphosphate, this is your chemical energy. Again, we put in glucose, and what we turn glucose into that powers our body is ATP. So ATP is real energy in our body. And so this is another type of a nucleic acid. It has nothing to do with DNA, RNA, and proteins and creating protein synthesis and all this stuff. And so this is a good example of how each one of these is a specialty molecule. Now there's a couple others that we're not going to focus on until we get to 211, some NADs and some FADs. You can if you want to. These guys are involved with ATP formation, and we'll talk about them as we move through into 211. Proteins, again, we're about to finish this up. We got about 8 more slides, but proteins, these guys are really they build the structure and they create the function. Okay, so whenever we talk about that, proteins, they build structure. I want you to make a note, building structure, things like muscle. Okay, so muscle, protein fibers. Okay, these are two very important things that we see. Muscle makes up a good bit of us, but also these protein fibers are found everywhere throughout the body. But they give us structure, support. And then also I want you to put function. And let's mention functional proteins, things like right here, enzymes, hormones, neurotransmitters. All of those create metabolic changes in their target cells. They make things change. And so this is how we function, is by using some of these proteins. So again, most of our A&P coming from these proteins. So these 2 semesters are really going to be a lesson in proteins, how they work and how they interact with other things in the body. So they have a vast array of functions, though. They can be catalysts to help make reactions happen, like an enzyme, be involved with defense, transport, support, movement, regulation, storage. So they've got a lot of different functions. Again, the building blocks, I didn't mention this yet, but the building blocks are amino acids. So the building blocks of proteins are amino acids, and a protein is simply just a beaded strand, and each bead is one amino acid. Now, we use 20 amino acids, and depending on the order we put amino acids in, we don't have to use all 20, but depending on which ones we use, in which order we use them, and how many we use overall, that creates a totally different protein. Our body can make hundreds of thousands of different proteins. So, pretty kind of cool. Proteins, these amino acids, we're going to bind them together peptide bond. If you ever hear that word peptide, that means linking amino acids, and anytime we see peptide, it also refers to proteins, because when we link amino acids, we're protein. So proteins are together with peptide bond between amino acids. Okay. Here we can kind of see some of these amino acids and proteins. Let's just move forward. We don't need to worry about that. As we get longer chains of amino acids, we can start to call them different things. To me, you have amino acids, then you have peptides. A peptide is a small protein, and then you have proteins, which are the larger proteins overall. So that's the way I like to think about it. Instead of talking about oligo and poly from 3 to 199, I just like to drop the oligo and poly, and I'll just refer to them as peptides overall. So we have amino acids are the individuals, clusters of them, smaller proteins we call peptides. The larger proteins, we just refer to them as true proteins. Okay. We can have other types of proteins with sugar attached, but I'm not too concerned about that. We will talk about that in the future. All right. So here's just a couple of diagrams wrapping it up. You can kind of see these. Here, this one's dealing with lipids in the textbook. You can also see this one dealing with carbs. And again, for some reason, half of my image is missing. And then here, dealing with nucleic acids, and finally with proteins. Now that kind of wraps up our discussion today of chemistry. So again, make sure that you're reviewing this. Hopefully you understand that we're focusing more of this information towards anatomy and physiology. We're trying to balance equations. You're not trying to predict and write out and draw all these different shells. We just want you to be familiar with the basic concepts here. So make sure that you're keeping up with any online assignments that you might need to be doing, and I will talk to everybody soon.